There we go. Okay. Hello, everyone. My name is Dominique Goodson, and I would like to welcome you to Warrior University session on gene editing, vectors, and sickle cell disease. Today we have as our presenter Dr. Julie Kanter, and she will be taking over from here. Hello, Dr. Kanter. Dr. Kanter, I think you're mute. Okay, there you go.
Okay, can you hear me now?
Yes, we can, and I'm going to give you presenters, giving you your presenter.
Hi, everybody, and welcome. Thanks for coming out or in or online, however you want to put it, on a Wednesday evening. Don't know what everybody else has been up to. I have been in clinic all day, and I'm excited to be here now. All right. I'm hoping that everybody can see the slides, but we are here today to talk about gene editing, vectors, and sickle cell disease. I am Dr. Julie Kanter, and I'm the director of the Adult Sickle Cell Program at UAB in Birmingham. These are the 4 pillars of Warrior University: research and education, best practices, legislation and policy, and life skills. And today we're really talking about research and education. Uh-oh, there we go. I hope you can see the slides again. These are a little better. So financial disclosures, I am required to let you know that I consult for a number of pharmaceutical companies and investors, all on trying to identify novel therapies in sickle cell disease. And I also have research funding and serve on several scientific advisory boards. Specifically today, I will mention the HGV-206 trial that is sponsored by Bluebird Bio, which is a publicly held biotech company based in Cambridge, and we'll be talking about Lentiglobin, which is their drug product. All right, so what are we going to talk about? We're going to understand the molecular basis of sickle cell disease. We're going to evaluate the different types of stem cell therapy for sickle cell. We're going to talk about gene therapy, gene transfer, gene editing, and are these all the same thing, or what is the difference? We're going to talk about what a vector means and how it works. And then we're going to talk about why we are even talking about gene therapy for sickle cell. So I know most of you all remember that sickle cell disease is a monogenic disorder. So it's caused by a single, one small problem in the genome that leads to the whole disease, where you have a mutation in the DNA that allows for a new protein to be formed, and that it's a single gene problem. Interestingly, first described well over 100 years ago by a student in the West Indies, sickle cell disease was also the first monogenic disease ever described, and the first one for which we understand the genetics is Mendelian genetics. Current therapies are limited and largely supportive, and of course rely on transfusion therapy, pain management.
I'm sorry. I apologize. Just one quick question before you continue. I just got messages that some people aren't able to hear you. Can anyone else that are having audio problems, please put it in the message. You may have to call in and use the number. Okay.
I can't.
Okay. Yes. Please dial in, everyone. If you cannot hear via your computer, dial in. Thank you. I apologize, Dr. Kanter.
No,
continue.
I can't. While I'm presenting, I can't see any of the questions. So
that's
definitely.
No problem. I'll make sure all the questions get to you.
Perfect. All right. So we were talking about that sickle cell disease is a monogenic disorder caused by a single gene, first discovered in 1910, and that our current therapies remain largely supportive. And those include transfusion therapy, pain management, hydroxyurea, L-glutamine, and obviously represent a significant health disparity. And that is Dr. Herrick down in that bottom corner. Just kind of cool that we have a picture of him. So it's caused by a mutation in a single gene, and as I mentioned, it's the first disease for which a molecular defect is identified. And so we know exactly what the cause is. We know exactly where the gene issue is. So why can't we just deliver a functional gene into the cells? Because this could reduce the symptoms of the disease. And again, we've now finally amassed the tools to consider that we might actually be able to do this. We might actually be able to deliver DNA into cells. Deliver DNA into a new cell to produce new hemoglobin in someone with sickle cell disease as a one-time cure. So this is a complicated slide that I'm going to take you through. Oops. Maybe. All right. So these are the different types of stem cell transplants that we talk about in sickle cell. The first is an allogeneic stem cell transplant, and HSC stands for hematopoietic stem cell transplant. A lot of people, especially those who don't know anything about blood cells, ask me, when we talk about stem cells, is this the same thing you hear about in the news? It's definitely not. These are stem cells that can only make blood cells. They're not stem cells that can make a foot or an arm or a leg. They can only make blood cells. So they're called hematopoietic, which means blood, stem cells. And allogeneic means other. So an allogeneic transplant is either from a matched sibling, a half-matched relative, also called a haplodonor, or an unrelated donor. Then we talk about gene therapy. This is where we take patients' own marrow, which we can actually use their actual bone marrow, or we can use blood that we gather from the periphery through pheresis, and we're going to talk more about that. We send the stem cells to the lab, and we insert a new gene. And that new gene encodes for new hemoglobin. That's gene therapy, otherwise called gene transfer. Then there's gene editing. So in gene editing, it's similar in that we take the patient's own stem cells again, but here we actually correct the misspelled beta globin. So we don't just add a new gene, we actually correct it. And I'm going to show you a figure that tells you what that looks like. Or instead of correcting the abnormal, where the mistake in the hemoglobin DNA is, we can use gene editing to turn on fetal hemoglobin. So as you remember, all babies make fetal hemoglobin at birth, but we have an off switch in our DNA. We have this off switch that can turn it off. And so gene therapy can also turn that back on. And then once we have edited stem cells, we can deliver those back to the patient. So why did we start this process? Well, we've known for a long time now that allogeneic bone marrow transplant is curative in sickle cell disease. I love my colleagues in Illinois, but they are not the first person to cure somebody for sickle cell disease with a bone marrow transplant. In fact, it's been done since 1996, when originally, actually, in earlier 90s, the first patient was cured on accident. It was a young girl who had AML. She had cancer. So she underwent a transplant and was cured of both diseases. After that, there was a study done where there was 22 children who were less than 16, and they were treated with myeloablative conditioning. Myeloablative means we really clean out all of the bone marrow, and had very high overall survival of 91%. But 18% rejected the marrow. That means when they got the new marrow, their body said, this isn't mine, I don't want it, and kicked it back out. And so some of those people continued to have sickle cell. However, learned at this time that myeloablative transplant is curative in children. France has some of the biggest and best results of transplant, and this is partly because insurance is so widely available in France. But they did this paper after they had 87 children transplanted. All of these children had a sibling, and the donor was either marrow or cord blood. And originally they used the same conditioning, but then they added this other agent, ATG. And that's important because after they added ATG, there was almost no rejection. All the marrow was really accepted into the patients, and there was an event-free survival of 95.3%. So it is curative 95% of the time in children with a sibling match transplant. And this was just a graph that looked at that. You can see the differences before and after using ATG. All right. So then the next group said, oh, well, that's great. We can do this in kids, but what about adults? So the NIH was the first group, and many have subsequently followed, including my friends in Illinois, to say, well, let's try doing a bone marrow transplant in adults. Here they used non-myeloablative chemotherapy, meaning they still used chemo, it just wasn't quite as strong because there was concern that adults had more organ dysfunction. And 9 out of 10 of those adults were able to engraft, meaning their bodies accepted the new bone marrow. It was very well tolerated. They normalized their hemoglobins, and they didn't have any further sickle cell disease or graft versus host disease. Graft versus host is important. That's when the new bone marrow starts fighting against the patient that it's in. And we really don't want to trade one chronic disease for another. All of these individuals demonstrated what was called mixed chimerism. What that means is they had some of their own stem cells and some of the donor stem cells. And with that, they were able to essentially normalize their hemoglobin and make far more normal hemoglobin than sickle hemoglobin. So then we had to ask the question, well, if they don't need to totally replace the stem cells, if they can have some stem cells that are still their own, how much new blood, new marrow is needed? And so we looked at several different studies, and there's one study that showed that non-myeloablative transplant performed in over 75 patients, and they looked at these 67 who had very long-term follow-up. And they looked at all the different ways we measured this donor bone marrow, including looking at hemoglobin and hemoglobin S, or the actual sickle production, and symptoms. And they found 3 patients who experienced declining donor engraftment. And what they found was that these 3 patients developed a rise in their sickle hemoglobin over 50%, when the new donor cells were less than 20%. So when you look at this, you can say, well, you need at least 20% of your new cells have to be from the donor. So 20%. The other 75 or 80% can be your cells, but 20% at least have to be from the donor. We also know a lot looking at people who have hereditary persistence of fetal hemoglobin. What does that mean? Remember, some people, well, everybody has fetal hemoglobin or baby hemoglobin when you're born. Some people, their switch doesn't turn it off completely. We understand that sometimes, but not other times. And so we know that fetal hemoglobin is protective, that it's the major genetic modulator that some people are lucky and born with. And so if people co-inherit this extra fetal hemoglobin, they often have very few complications of their sickle cell. One study, when you looked at how much fetal hemoglobin do you need, showed that for major organ failure, you need to have at least 10%. But for recurrent clinical events like pain crises, you have to have at least 20%. And more recent data suggests about 30%. So you need a lot of fetal hemoglobin to prevent some of these problems. So it's also important to learn how the red cell works, because there's a lot of people, there's certainly almost every med student who don't understand a red blood cell. So what's important to know about a red blood cell is that a normal red cell has lots of hemoglobin in it. It's not just 1 or 2 hemoglobin molecules, it's tons. So a normal hemoglobin AA red cell has all A and a little bit of A2. Somebody who has trait, there we go. Somebody who has trait, who inherits 1 A from one parent and 1 S from the other parent, in every single red blood cell, they have a mix of hemoglobin A and sickle hemoglobin, every red cell. And that's why they don't have any symptoms, because in every red cell, there's more A than there is S. And so the cell never sickles, and they never have that inflammation that people with sickle cell disease have. If you have SS disease, all you make is hemoglobin S, with just a little bit of A2. And so your cell is filled with SS. Well, what if you co-inherit that fetal hemoglobin like we talked about? When you co-inherit fetal, that fetal hemoglobin, similar to trait, is in every cell. And again, the more fetal there is, the more protected that cell is. Now that's different than people who are on hydroxyurea. In hydroxyurea, you have all SS cells or all FF cells. So some cells have all sickle and some cells have all fetal. This is very important because it can really predict how much efficacy we're seeing. So when we think about it with a more mild, if you look all the way to the right, or more asymptomatic sickle cell disease, that in every cell they have more fetal hemoglobin, and there's more than what we measure as 10 picograms. If they have moderate phenotype, they have some fetal hemoglobin in every cell, And if they have a more severe phenotype, they have almost no fetal hemoglobin. So it's really important, looking back at this slide, that we want, when we start talking about gene therapy or making new cells, we want all of our patients after transplant to either look like the top left, where they make AA cells, the bottom left, where they make SF cells, or that they look like they have trait, so that they have a mix of the non-sickling hemoglobin and the sickle hemoglobin in every red blood cell. So what do we know? Stem cell transplant is absolutely curative for sickle cell disease. You need at least 20% of that donor bone marrow to stick in the inside to correct sickle cell. And that low toxicity means that we can do this in adults, but that most patients still don't have a donor. And so we have to look for alternative stem cell sources. So there's unrelated or cord blood donors, but we've also found in the United States that less than 10% of people with sickle cell disease have an unrelated donor. And they're haploidentical or half-matched transplants. And some of these outcomes are amazing and getting better and better, but they still involve immune suppression and the risk of graft versus host disease. So we're still looking for other options. So the question is, can we achieve the same 20% with gene therapy if we use the patient's own cells? So when we talk about using their own cells, we harvest them first. And there's two ways to harvest stem cells. The first is through the bone marrow, where we put a needle in the back and withdraw the cells. Now, patients are asleep when we do this, so don't worry about that. But we don't put the stem cells. And so the other way to do that is through a pheresis machine, where we put in an IV, connect the patient to a machine that sorts their cells. It's like the coin counter at the grocery store. It only takes out the stem cells and gives them the rest of their cells back. Once we have those cells, we can send them to a lab where we can add a new gene, a correctly spelled beta globin gene, using the viral-based transfer. The patient would then get chemotherapy that helps them make room in their body for the new stem cells with that new gene, and they get transplanted. Okay, now, elephant in the room. In this case, it's a Trojan horse in the room. What about these viral vectors that everybody's heard so much about? So we're going to talk about the Trojan horse. This goes back to the story of the Greeks and the independent country of Troy many, many years ago. If you don't know the story, the Greeks were trying to get inside the independent nation of Troy that had this amazing wall built around with a gate. And they couldn't get in, so they were trying to take over Troy, but they literally couldn't get into the country. So as a last-ditch effort, they actually built a horse that looked like a gift. And it was huge. And the Greeks, the soldiers, hid inside this horse. And the people of Troy said, wow, they left us this gift. They've given up. They withdrew. When they looked out in the ocean, there was no ships anymore. And they let in this horse. And then the Greeks jumped out of the horse and killed them. And that was an actual reimagined picture of what that horse looked like. Those are my imagination of the soldiers. So we're going to talk about the Trojan horse as our virus. Looking at the left side, when you think of the Trojan horse, it has two parts. It has the wooden horse, and then it has the viral bad stuff on the inside. And so when you start thinking about a vector, what we do is actually take out the viral bad stuff. We take out the soldiers outside of the horse, but we leave the horse. And now it's an empty horse, but it still looks really good, still looks really cool, and Troy still wants to bring that horse inside the gates. What does that mean? So now we're going to think of it the same way, but we're going to talk about it as a viral vector. So that empty horse, right, it's got nothing in its belly. We're going to repackage it, and we're actually going to put in the new gene. The edited gene is going to go into that viral vector. And in the case of the study that we talk about most lately, that horse is a lentiviral horse. So it's made from the same virus that we make HIV from. It's actually a parent of HIV, but it's the lentivirus. But now we've taken out the yucky part, taken out the soldiers, and just left the wooden horse. And we package it with the new gene. And interestingly, we put a bow on it, and that bow comes from a totally different virus. And then between the horse and the bow, this stem cells say, wow, that looks pretty good, we're going to let you in, and lets the horse in. And once the horse and the bow are in there, they empty out the new gene into the cells. And the new gene puts itself into the DNA of the new gene. Now, the horse can't make copies of itself. Unlike a virus that's activated, this horse can't do anything. It had all its M sides emptied out. So the cell eventually just spits it out and it dies. It's all gone, and all that's left is that new gene. So this is what the clinical model of beta globin looks like. This is the actual structure that they put the new gene in, and that new gene is that beta T87Q. This is the new gene that encodes for healthy hemoglobin A. So the viral vector, the horse, can't make copies of itself after it's in the new cells because it doesn't have any of its machines. It's been emptied out. And the new cells, the stem cells use their regular sort of machinery to replicate themselves and make new copies of this gene, the globin gene. And that allows for people to make healthy hemoglobin or beta hemoglobin or hemoglobin A. Those are all the same thing. Now, let's take a break from gene transfer and talk about gene editing, because they are truly different things. So scientists can use gene editing, and you may hear it called CRISPR-Cas9. And it's not just used in sickle cell disease, it's used in other places. And what they do is they design an RNA segment to exactly look like the DNA segment they want to target. And they then use another virus to insert that DNA segment along with some chemical scissors, and that's called Cas9. And it goes in and it cuts a place to unravel the DNA, and it matches right where they have lined it up because they have made this very perfect match. And then one of two things can happen. They can either just cut it and let the cell heal itself, and when the cell heals itself, it doesn't heal perfectly, or they can also add in a new piece of DNA. So what does that mean? So this is just another picture. When we talk about that CRISPR-Cas9, you can see the blue DNA, and that pink is the guide. That pink is what the scientists have designed to exactly match the target area that they want to use. Now they connect the chemical scissors to it with the Cas9 enzyme. And so it knows to target right here. It's only going to bind up this DNA, and it's going to cut. And then it leaves a hole. And now two things can happen. They can let this repair naturally, and it almost doesn't ever repair correctly and develops what we call indels, or they can insert another piece of DNA. Now, why would you do these different things? Well, if you're trying to turn on the off switch for fetal hemoglobin, all you need to do is disrupt that off switch. And if you disrupt it, it turns it back on. You don't need this extra DNA. But if you want to go in and actually cut the mutation that causes sickle cell right at that place, you want to cut it and then repair it. You want to put in that healthy hemoglobin DNA so that it will make exactly what you want it to. I apologize, this is a little blurry, but this is another version of those two different techniques I just explained. The same idea up here at top, where you have this perfect guide and this chemical scissors. In one way, called non-homologous end joining, after they've made this chemical cut and there's a break in the DNA, they just let that DNA repair itself. And it gets what we call indels. And what we're trying to do here is to turn on and off switch. The other option is called homologous directed recombination. Here, when they have this special scissors, they add in a piece of DNA. So once it makes that cut, this extra piece of DNA goes right in there because it fits perfectly. It's like the missing piece. It fits perfectly, and then it will make the right DNA. And this is how you can actually directly edit cells. Either way, whether we've done gene therapy or gene editing, the same thing takes place. We take stem cells from the patient. Now we use apheresis to collect those cells. We put the cells into a lab, and we transduce them. That means we use a virus. We use that empty horse to send in the new contents that we want in. When our cells are all ready, we give chemotherapy that makes room in the bone marrow for the new cells, and then they're transplanted back. So these are some of the results from the gene therapy study HGB-206, because this is the study that's furthest along, and we can tell you a little bit more about it. So when they first did this study, when they collected stem cells, we used a bone marrow harvest. And we found that the cells weren't great, and that's a whole nother discussion. And we gave chemotherapy, and then we transduced the cells, and we infused them back. We also didn't require transfusions up front. And we used the original manufacturing process. So when we sent those cells to the lab, and they did their special sauce to make the horse go into the cells, they used what they call the original process. And we didn't get the levels that we wanted. So they changed it. And we started in Group B using a refined manufacturing process. So we gave it new special sauce, and that made far more horses deliver the gene to the new cells. And then we did plerixafor mobilization, where we used pheresis instead of bone marrow harvest, and we required everybody to be transfused. And all of that together led to better outcomes. So what we saw, and one of the ways that we count or see how well things are going, is we measure vector copy number. In other words, we measure how many horses are in the cells. And in our first group, we saw not as many as we wanted, less than one horse per cell. We also saw that instead of 100% of the stem cells getting the horses, only upwards of 40% of the stem cells were getting those horses. So now only 40% are getting the horses at all, and of those cells getting the horses, there's less than 1 per cell. And that wasn't what we wanted to see. So when we made improvements using that refined process, the new special sauce, now we're getting 3 or more horses per cell, and we're getting 80 to 100% of the stem cells with horses. So remember, we want a minimum of 20%, preferably more, so 80% was much, much better. And then when we looked at, finally, in our Group C, or our most further along group, you can see that those were sustained, that we continued to get more horses per cell. We got more of the cells total with the horses in them. And because we were using the new collection process and not harvesting bone marrow, we were getting a lot more cells from each patient. All of that together led to much better outcomes, and this is just looking at the different number of harvests people had to undergo, what that drug product vector copy number was, and how many transduced CD34 cells. So this is just the same words that I just showed you before in pictures. And what does that translate to? This is hemoglobin. So what we're looking at here is the green is hemoglobin S. The pink is that healthy T87Q hemoglobin A. The yellow is baby hemoglobin or fetal hemoglobin, and blue is the transfused. And you can see in our first few patients in Group A, they all made some pink, some T87Q, but there's a lot of green. And then as you can see, as we moved into the Group Bs, they're making 30 to 50% healthy hemoglobin. And I don't have the graph up here, but we showed it last year. As we moved into Group C, everybody's making above 40% healthy T87Q. When we looked at the outcomes and the safety, it was very safe. Everything we saw was related to the chemotherapy itself, and there was no replication-competent lentivirus. In English, that means just like we planned, those horses could not make copies of themselves. The horses all went away, and that was very important. And when we looked long-term, we could see that the patients who were in that later group in B and C continued to have the number of vectors that they were supposed to have and were making a lot of good, healthy hemoglobin over time. And this was one of our patients who at 6 months out had 75% of the transduced cells. So he had 75% of all the cells in his marrow had the horses in them. And again, that was really important because we needed at least 20%. And that told us that we were making headway towards a curative procedure. So when we looked at this study over time, we saw that Group A, even though they only made some of the healthy T87Q hemoglobin, actually still had clinical benefit. But that the early data from Group B and then later from Group C really showed us that all these changes made it an improved process so that we were getting 50% of total hemoglobin right where we wanted it to be of non-sickle hemoglobin. So I'm going to stop there so that I can answer any questions and we can talk about it.
So does anyone have questions? Please remember, if you have a question, to unmute yourself. I just checked the chat box. There are no questions. So if anyone has any questions for Dr. Kanter right now, please just unmute yourself and go ahead. Thank you.
All right, I see one question. What strategies do you use to explain these technical details to patients and families? I actually use a very similar strategy to what we just talked about. I either get out the slides of the horse, because people really understand that, or I talk a lot about, my favorite analogy is a letter and an envelope. People really can understand that if a virus is made of a letter and an envelope, and you take out the letter that causes you to be sick, you take that out, rip it up, throw it away, then the envelope still gets to where it's going, and we use another virus to put a stamp on it. So the envelope is made of one virus and a stamp is made of a second virus, and the new gene goes into the envelope, and it gets delivered into the cells. And that's something that people can really visualize and understand. Oh, good. I'm glad that helps.
Are there any more questions? Oh, we have one.
What range of health literacy do you see in patients and families? It's a very, very wide range. I see everybody from, parents or patients who've not finished high school, some who never finished middle school, to those who are, master's and graduate level educated. And it certainly changes the way I talk, because some people don't want me to talk what they consider talking down, and other people, want to just be talked to like a, quote, normal person, even if they're a doctor. And so I try to use very similar language, but it probably does change a little bit on the words that I use. In terms of study location, the gene therapy study, the one I just talked about, is multi-center. There are currently, I think, 7 or 8 sites in the United States and a few internationally. And there's some gene editing sites as well that are across the country. So all are multi-center. What questions did you hear following 60 Minutes? I think everybody on 60 Minutes didn't hear anything past HIV. I really think a lot of people got stuck on HIV and didn't get to hear anything else. So I got a lot of questions about, did you say HIV, or did you say you were giving sickle cell patients HIV, or you're just experimenting on people with sickle cell disease? I think that was the majority. And then, several people asked other questions like, did it work? Or how do people feel now? And they were all really good questions. And I think it wasn't well explained on the 60 Minutes. So I actually put something on my research page that really tried to explain that the HIV is the empty horse, and it can't make people sick. And not only are we doing this in sickle cell, but we're doing the exact same thing in thalassemia. And in fact, in thalassemia, they've transplanted 3 times as many patients. So we have a lot of data that this vector is very, very safe, and we haven't seen any problems. I got questions about the patient who has myelodysplastic syndrome. So one of our very first patients on the study developed a side effect or a complication called myelodysplastic syndrome, and it's precancerous. And he actually has to undergo another transplant now. And people wanted to know, was that from the virus or was that from the transplant? And we did a lot of investigation to figure out that the virus was not involved and that the cells that caused the myelodysplastic syndrome didn't have any horses in them. So we actually knew the virus had nothing to do with it, and that it was probably the combination of chemotherapy plus years of hydroxyurea.
Do we have any more questions for Dr. Kanter?
Well, certainly, if anybody has more questions, feel free to reach out to me, shoot me an email. You can message me on Facebook. Any of those things work, and I will be sure to get back to you. All right, are we good?
If anyone doesn't have any other questions, you got a lot of thank yous for clarifying the different vectors, which we all appreciate very much because, as you said, a lot of people for the 60 minutes after hearing the word HIV, no one heard anything else, just the word HIV. So thank you so much for explaining this and helping us understand more about this whole process. So thank you so much, and we appreciate it.
All right, my pleasure. And as I said, feel free to reach out if you need anything.
Yes. So if you guys don't have any more questions for Dr. Kanter, we're going to call that a session. Thank you guys for participating. And if you have any questions like Dr. Kanter has said, you can reach out to her or reach out to the consortium, and we could help answer your questions or get your questions over to Dr. Kanter. And we will be having more classes on gene editing in the future for Warrior University. So make sure you stay tuned if you want to learn more about it. Okay? Thank you all for participating. Thank you so much, Dr. Kanter. This was awesome, and we loved it.
Good. I'm so glad. Take care.
Yes, thank you. You too.
Bye-bye.
Have a great night, everyone. Thank you.